Hello everyone, my name is Philip Fradkin, and today I'm going to be talking about CONCERTO, a graph neural network approach for molecule carcinogenicity prediction. And this work was done with my fellow co-authors and PhD students Adamo Young and Lazar Atanakovich, and it was supervised by Drs. Brendan Fry, Leo G. Lee, and Bo Wang, which was done at the Tsai and Wang labs at the University of Toronto and the Vector Institute. So first, I'm going to introduce carcinogenicity as a molecular phenotype. So we can sort of think about carcinogenic compound discovery in two modes. The compound can have either a really high effect size, or there can be a population with high exposure to this compound. So on the left, we have this plant called birthworts, which contains aristolochic acid. And in the early 2000s, this plant was used as a weight loss treatment, and it was found to be extremely carcinogenic, as aristolochic acid is extremely potent. And on the right, we have this population of chimney sweepers, and in 1775, Percival Pott identified this group of people, to have high rates of squamous cell carcinoma. And it was later discovered that this is caused by benzopyrene, which is in high concentration in soot. So what about compounds with intermediate carcinogenicity and intermediate exposure? So we would like to be able to identify those, and an effective approach would increase the percentage of positives identified, so increase the number of carcinogenic compounds, and it would also be low cost and high throughput. So in this work, we propose an in silico compound selection approach for downstream carcinogenicity experiments. So there are two sort of main approaches to measuring carcinogenicity. There's the carcinogenic experiments, which are usually done in rodents, where a group of animals is exposed to the compound of interest, and then tumor growth is observed. And then there are mutagenic experiments, where you have a cell culture, and this culture is exposed to your compound of interest again, and then a molecular phenotype usually measured is cellular growth. So carcinogenic experiments, they measure genotoxic and non-genotoxic effects. And this just refers to the method of action for the compound carcinogenicity. So genotoxic refers to DNA damage caused by the compound. So as an example, alcohol is considered non-genotoxic, but is carcinogenic. And on the plus side, these experiments measure the true phenotype that you're interested in, and they measure different kinds of carcinogenicity effects, but these experiments are often expensive and quite slow. And on the right are mutagenicity experiments that measure only genotoxic effects, but they're relatively cheap and so are very abundant. However, these experiments have lower rates of replication. So at its core, carcinogenicity prediction is a graph-level regression problem. And there are a couple of core challenges associated with this. So first of all, carcinogenicity prediction by itself is extremely data constrained. So there are only about 1,000 samples available with high quality level readouts of carcinogenicity dosage, compound carcinogenicity effects. And then, as I mentioned, it's actually a continuous phenotype. So a lot of previous works framed it as a binary prediction challenge. Either a compound is carcinogenic or not, but it's actually a function of dosage. And finally, this is actually a very complex phenotype. So there are multiple molecular mechanisms going on, and unfortunately, we're currently unable to tackle this challenge because it requires higher detail level of data. So now I'm going to talk about some of the contributions of our work. So first, we interpret carcinogenicity as a regression problem, which results in more informative labels. We pre-train unrelated but lower quality mutagenicity experiments prior to predicting carcinogenicity effects. We conduct transfer learning from a large self-supervised model, and we're the first to our knowledge to apply graph neural nets to carcinogenicity prediction. And finally, for model interpretability, we generate counterfactual molecules for model interpretation. So there is a variety of ways that you can represent a molecule. So on the left is a graph representation, so where atoms are nodes and edges are bonds between the atoms. In the middle, it's a string representation, often refers to SMILES, and there is a selfies format and a couple of others. And this format has the property of many to one mapping. So that means many strings can refer to the same molecule. So in theory, if we have a computational approach, we would want it to be invariant to how we represent the molecule as a string. And then finally, we can represent it as fingerprints, which is a binary vector representation. And fingerprints are bitwise vectors indicating molecular subgroup membership. So whether a given substructure is present in the overall molecule. And it can be thought of as a hand-engineered approach to molecular representation. So we utilize 3 data sets, 2 carcinogenicity and 1 mutagenicity data set. And the mutagenicity data set is an order of magnitude larger. So we have about 6,000 molecules in it. And unfortunately, only one of our carcinogenicity data sets has continuous measurements, which is TD50, a measure of tumor count normalized to dosage as a proportion of animal body weight. So this is the overall diagram of our model. So we have in blue the Grover gene and transformer. In green, we have an MLP fingerprint predictor. And then in orange, we fine-tune this representation with multi-round mutagenicity pre-training. So Grover is an 100 million parameter model trained on a 10 million molecule dataset by Yu Rong et al. And published in 2020. And at its core, it's a graph neural net, which the output of which is then fed into a number of transformer layers to learn a local neighborhood representation as well as a global molecular representation. And it's a model that's self-supervised and is trained in an unsupervised way using masked language modeling techniques. So in green, we have a concatenation of this molecular fingerprint vector as well as the Grover representation, and it's passed into a multi-layer perceptron. And this architecture contains individual carcinogenicity and mutagenicity prediction heads. And then finally, this representation is trained, it's pre-trained on this lower quality mutagenicity data that I mentioned earlier, which is a classification. And then the model is trained on carcinogenicity data as a regression task. And we find that actually if we iterate over these objectives, empirically the performance is better. So we repeat this process multiple times. So overall, the full model is trained end-to-end using backpropagation. And an additional point is that Grover weights are actually frozen, which is a common convention for fine-tuning these large models. So now I'm going to talk about our results. And the full model does quite a bit better if we compare with just the fingerprint component or the Grover component. So concatenation of the two representations improves the continuous performance on the test set by quite a bit. And we also outperform previous carcinogenicity prediction models, such as carcinopred and the AdaBoost model from Limbo et al. In addition, we find that just the Grover prediction by itself is actually quite variable on the continuous prediction task. And then on the held out test set, carcinogenicity test set, we find that CONCERTO as well outperforms the previous models, achieving a higher ROC area under the curve. And then we were interested in investigating the role of mutagenicity pre-training. And we trained a series of models that either only do one round of mutagenicity pre-training versus multiple rounds of mutagenicity pre-training, and find that increased mutagenicity pre-training improves performance. So finally, this is, I find, a very interesting part of our work. We would like to understand better what the model is actually learning. And to do that, we look at counterfactual molecules. And so the goal is to find a sample that is close in the input space, but far away in the prediction space to a sample of interest. And it's done by, given a molecule of interest, we do a random walk in the string space representation of a molecule, and we consider Tanimoto scores as a way of measuring molecular distances. So here, this is a plot representing how these counterfactual examples are found for a single molecule. So here in black, we have our molecule of interest, and that's indicated by a distance of 0 to the original molecule. And then we're randomly mutating the structure of the molecule, and are interested in identifying the lowest distance to the molecule of interest, which is on the x-axis, but highest change in predicted carcinogenicity, which is plotted on the y-axis. So for example, given our base molecule in blue plotted, we remove the aliphatic iodine group, and we think that the model thinks that addition of a hydroxyl group reduces the predicted carcinogenicity of the molecule. And then addition of a nitroso group increases the predicted carcinogenicity of the molecule. And we identify the red molecule as the positive counterfactual of our molecule of interest. So a molecule that is similar in structure, but is significantly different in terms of its carcinogenicity prediction. And then we can expand this analysis dataset wide. So for every molecule in our test set, we can identify a positive counterfactual and a negative counterfactual. And then in prior work, researchers have identified substructures, molecular substructures that are associated with carcinogenicity. And then given these substructures, we can count what's the percentage of the time these are found in molecules, in the original molecules in our test set, in the negative counterfactuals that the model thinks should be less carcinogenic, and the positive counterfactuals that the model thinks that should be more carcinogenic. So for example, nitroso group, which is considered, which is identified to be more common in carcinogenic molecules, it's present in 21% of our test set molecules, but it's present in just 3% of our negative counterfactual molecules. So when we ask the model to return a molecule similar to the one in structure, the model often disrupts this nitroso group, removing it. And on the other hand, when we ask for a model to return a more carcinogenic molecule, then this nitroso group is found in 32% of our positive counterfactuals. And we are able to observe similar trends across these other toxicophore, groups that are identified to be carcinogenic by prior work. So overall, we think that molecular carcinogenicity prediction is an important problem, and it's especially important to think about compound dosage while assessing model performance. And currently, our performance doesn't achieve levels needed for clinical decisions, but we hope it can be used for selected molecules for downstream carcinogenicity experiments. And then more broadly, we find often in life sciences, we encounter these data-constrained problems. So a few general techniques that can help are large models pre-trained with self-supervised learning, then pre-training on related experiments, even if they could be lower quality. And then finally, encoding domain expert knowledge, for example, with fingerprint vectors. But overall, still the most effective way to improve performance is to acquire more data. And on this front, we're sort of optimistic because CPDB, which is the carcinogenicity dataset, was updated last in 2007, and Hansen mutagenicity dataset is from 2009. So I'm sure that there is additional data out there, and if you're interested in collaborating or retraining the model on a larger set of data, that's something that would be very excited to do. So Thank you so much for listening to my talk. And if you have any questions, please reach out, and I'd be happy to chat. Thank you very much.